Lymphocytes activated by antigen give rise to clones of antigen-specific cells that mediate adaptive immunity. The defense systems of innate immunity are effective in combating many pathogens. They are constrained, however, by relying on germline-encoded receptors to recognize microorganisms that can evolve more rapidly than the hosts they infect. This explains why they can only recognize microorganisms bearing surface molecules that are common to many pathogens and that have been conserved over the course of evolution. Not surprisingly, many pathogenic bacteria have evolved a protective capsule that enables them to conceal these molecules and thereby avoid being recognized and phagocytized. Viruses carry no invariant molecules similar to those of bacteria and are rarely recognized directly by macrophages. Viruses and encapsulated bacteria can, however, still be taken up by dendritic cells through the non-receptor-dependent process of macropinocytosis. Molecules that reveal their infectious nature may then be unmasked and the dendritic cell activated to present their antigens to lymphocytes. The recognition mechanism used by the lymphocytes of the adaptive immune response has evolved to overcome the constraints faced by the innate immune system and enables recognition of an almost infinite diversity of antigens so that each different pathogen can be targeted specifically. Instead of bearing several different receptors, each recognizing a different surface feature shared by many pathogens, each naive lymphocyte entering the bloodstream bears antigen receptors of a single specificity. The specificity of these receptors is determined by a unique genetic mechanism that operates during lymphocyte development in the bone marrow and thymus to generate millions of different variants of the genes encoding the receptor molecules. Thus, although an individual lymphocyte carries receptors of only one specificity, the specificity of each lymphocyte is different. This ensures that the millions of lymphocytes in the body collectively carry millions of different antigen receptor specificities, the lymphocyte receptor repertoire of the individual. During a person's lifetime, these lymphocytes undergo a process akin to natural selection. Only those lymphocytes that encounter an antigen to which the receptor binds will be activated to proliferate and differentiate into effector cells. This selective mechanism was first proposed in the 1950s by McFarland Burnett to explain why antibodies, which can be induced in response to virtually any antigen, are produced in each individual only to those antigens to which he or she is exposed. He postulated the pre-existence in the body of many different potential antibody-producing cells, each having the ability to make antibody of a different specificity and displaying on its surface a membrane-bound version of the antibody that served as a receptor for antigen. On binding antigen, the cell is activated to divide and produce many identical progeny, known as clone. These cells can now secrete clonotypic antibodies with a specificity identical to that of the surface receptor that first triggered activation and clonal expansion. Burnett called this clonal selection theory. And that's lymphocytes activated by antigen give rise to clones of antigen-specific cells that mediate adaptive immunity. Have a great day.